Hello friends. In the previous videos, I have explained about the Selaginella. So in the Selaginella, I think you are observed the one important characteristic, that's a Heterosporus condition. That means usually the Selaginella, that's a pteridophyte, produce the more than one kinds of spores. That special characteristic of Selaginella is referred as Heterosporus condition. So in the today's video, I will be explaining the topic on Heterospory and seed habitat. Usually, what is Heterospory? Usually the most of the pteridophyte produce the only one kind of spores. They are referred as homosporous. Homo means single or one type of the spores. But especially in case of the Selaginella or some of the pteridophyte, so they produce the more than one kinds of spores. They are referred as the Heterosporus. So what is a heterosporus? Hetero means different. The size may be varies with those spores. That's the microspores and the megaspores. So usually the microspores produce in a structure called as microsporangia, and the megasporangia produce the megaspores. So they have the different size in the spores. So usually microspores germinate to give rise to the male gametophyte, and the megaspores germinate to give rise to the female gametophytes. So that's why they different in the size of the spores related to the sex of the gametophyte. So that's why they are called as Heterospory. That means structurally, functionally different spores produced in case of the some of the pteridophytes. They are referred as Heterospory, and the condition is known as Heterosporus condition. So especially in case of the Selaginella, Marsilia, Salvia, Azolla, these will have this special characteristic. So coming to the different theories has to put forward to explain the Heterospory nature. So the study of the Heterosporus originated due to the reduction in the number of the spores within the sporangia. Sporangia is a spore producing cavity. In that, the spores will be reduced, and when the spores of sporangium are functionally, they have the spores is recessive and limited food supply will be there. That time what happens, usually small spores will be produced, and during the when the spore mother cells in the sporangium disintegrate during the development, they remains want to get the sufficient nutrients. So that's why sometime maybe they will be reduce the size, or sometime maybe enlarge. When they get reduced or remain smaller in size, they produce the microspores, and they will larger size, the numbers will be less, they are the megaspores. So usually various evidences, that's the paleobotanical and developmental experiment, shows that these are the some reasons to development or formation of the microspores and the megaspores. For that, various scientists has explained why these heterosporous conditions will be formed. So based on the some evidences from the paleobotanical studies, we have gathered the knowledge of the heterosporous condition. First one is to Scott in 1894. He has explained the two spores of the, that is a Clambostachys, that's a Clambostachys biniana and Clambostachys cashiana. So he explained the spore size will be reduced and the large number of spores will be produced. So that is the because of the some developmental stages of these spores have the nutritional deficiency. But in case of some certain species of the Lepidocarpon, they may be varies in the size and the shape of these spores. Second one is the evidence from the development of the structure, that's a developmental stage, that's how actually the cells will be developing. So that's a sporocyst and meiosis, and also the maturation of spores, they will support the these heterosporous habitat. And some of the other condition, that's abortination, and that will also lead to the formation. Thus was explained by Rashid in 1976. And similarly, in case of salvia azola, also explained by various scientists. And third one is to from the experimental results. So Selaginella and Marsilea especially, they focus on the nutritional factor. They will lead to the formation of this heterosporous condition. Sometime in case of the photosynthetic activity, that will also lead to the heterosporous condition, or that will lead to the heterospory expression of the sex determination capacity. So these are the some important reasons for the heterospory. One is heterospory express sex determining capacity of the plant. In the heterosporous species, differentiation of the sex takes place at the gametophytic stages, whereas heterosporous species differentiate in size, spores in relation to the sex of the gamete. Second one, an important characteristic of seed habitat is that megaspore is retained by the parent even after the fertilization. This ensures nutrition for the developing embryo. And third one is to biological significance of the heterospory is that the heterospore form development of the gametophyte in the endospores, and the nutrition for the developing gametophyte is derived from the sporophyte. The nutrition is derived from the that is usually the sporophyte. Hence the development of the gametophyte is not affected by the ecological factors in case of the independently growing gametophyte. This is about the heterosporous condition and the seed habitat. Once the spore forms, how actually the seed will be there. In order to evaluate the seed habitat, so first you have to be study the situation of the seed bearing plant, that's a spermatophyte. They have the spermatophyte two types of spores, that's a micro and the megaspores. Usually microspores are the smaller grains and megaspores are the ovules. The male and female gametes to fuse form a zygote, and that zygote will be develop lead to the formation of the embryo inside the ovule. The embryo and the rest of the gametophytic tissue, that's intimate and ovary walls to formation of the help in the formation of the seeds. There are two types of spores in case of the Selaginella or some pteridophytes, that's a micro and megaspores. They are the heterosporus. During that time, what happens, so some modification will be occurs and retention of the megaspores in the megasporangium until the development of the embryo, elaboration of the megasporangium, and availability of the sufficient nutrient to develop the embryo. So based on that, evaluation of the seed habitat in case of the pteridophyte can be done. One is to most common genus, that's a Selaginella, have these types of heterospory and seed habitat. However, the seeds will be developed with the some modification, that the megasporangium lacks integument and permanent retention of the megaspore within the megasporangium. After development of the embryo, the resting period is not there. All these purposes, the seed habitat will be modified and will lead to the formation of seed habitat. So this is the how the actually heterosporus and the seed habitat both have the correlated. That means the if the heterosporus condition, generally what happens, one larger size of the spores will be there, that will lead to the formation of seed habitat or seed bearing structures in the plant. So that's why in case of Selaginella or Marsilea, usually that with the seed habitat. In case of the Lycopodium, you will find only spores. The small spores will be, or uniform homospores will be there. But in case of Selaginella or Marsilea or sometime maybe Azolla, you can find the small different size of spores, and also that's the micro and megaspores, and they have the seed structure will be there. So that's why the concept is referred as Heterosporus, spory and seed habitat characteristic. In the examination, you may have to get a general explain the heterosporus condition in case of Selaginella, or just general account, write the significance of heterosporus condition, what is heterospory, where do you find heterosporus condition, give an example. So like that, so many questions will be asked in the examination. I will get the concept of heterosporus and seed habitat characteristic. So if you have any doubt regarding this concept, so you can ask the question in the next Google Meet classes. But from the exam point of view, this heterosporus condition is very important. This is how actually heterosporus, the two theories has to put forward, are the three evidences, that is paleobotanical evidence, experimental evidence, and the developmental evidence, lead to the formation of the heterosporous condition that has to be explained by taking the example of the Selaginella and seed habitat also explained. Just if generally directly asked for seed habitat, only 2 marks question will be asking, but heterosporous condition will be asked for 5 marks question, 4 to 5 marks questions. Okay, thank you for patience listening.